Hey everyone, we are continuing chapter 14, nervous tissue. We're starting here on slide 13. Chapter 14, slide 13. We've talked about the basics of neuron anatomy and the glial cells that we find in the peripheral nervous system versus the central nervous system. Here we're going back and focusing on an individual neuron and how its individual parts help it spread an electrical signal. If we look at let me draw a basic neuron. This spiky area, I'm trying to draw the dendrites. Here's the axon, hillock, and axon. And then an axon terminal. So dendrites, where the nucleus is, that's the cell body. Axon, hillock, axon, terminal. Dendrites detect. How they detect things, it depends on what type of neuron it is, and we'll get to all of that later. But let's say we're detecting a signal from another neuron. So what we're looking at right here is the axon terminal of another neuron. So that's what we see here. The axon terminal of a different neuron sending a signal to the dendrites and cell body of the neuron that we care about. So let's say from this axon terminal, we get a release of a chemical. This chemical is known as a neurotransmitter. Use a different color. A neurotransmitter are these chemicals that get released into the synaptic cleft. Neurotransmitters are released by an axon terminal into the synaptic cleft. When those neurotransmitters reach the dendrites on their plasma membranes, dendrites have receptors. We've seen this idea before with neurons controlling muscle. Neurotransmitter is released from the axon terminal, goes across the synaptic cleft, binds to a receptor. When the receptors are activated, they open up sodium channels. Na+ is sodium ion. Again, we've seen this before. Sodium rushes in, and now we get a spread of sodium into the cell body. Let's assume that only one channel was opened up. We get a spread of sodium into the cell body. Let me point out the sodium channel. So sodium starts coming in. Remember, sodium has a charge. It's a positive charge. So we're getting positivity, positivity into this area. The closer you are to the channel, that's right where all this sodium ion is coming in, it's going to be pretty positive here. There's going to be a lot of positive ion here. The farther you get from that channel, it's going to spread out more. So there's less here, there's less here, there's less here, there's less here, there's less here. So there's more by the channel, and then you get gradually less. Another word for gradually, another word that you can use to describe this is that there's a gradient. There's a gradual change from lots of ion to less ion. Another word for electricity is a potential. So this is a graded potential. We call this a graded potential. It's very positive right where the ions came in. It's less positive farther from where it came in. In other words, it has a gradient, a graded potential. It's kind of like why I have this picture or this animation on the right side. If you ever play that strong person competition at the fair, you take a hammer, your stimulus, and you hit the target. When you hit the target, you can see a light or something go up. Depending on how hard you hit it, that light might go up a little bit if you hit it soft, it goes up more if you hit it harder, it goes up really high if you hit it really hard. There is a gradation depending on how much stimulus you get. How much sodium comes into the cell, it depends on how much our cell was stimulated. Depends on how many times this dendrite was stimulated. That's how we get a graded potential, this flow of sodium ion, this flow of electricity into the dendrite cell body side of our neuron. Great, so now we have electricity in our cell, but what do we do now? This isn't the main signal that goes down the axon. We haven't gotten there yet. We haven't made a decision yet. This is just the sensation. This is just the input. So again, here's the axon terminal of one neuron. Here's the dendrites and cell body of the other neuron that we're looking at. If we look at that electricity, that graded potential, remember it's very, very positive in this area closest to where the channel opened up. It's not going to be as positive out here where the axon hillock is. It's just not. It's never going to be as strong. But if we get enough of these sodium ions to flow to the axon hillock, if we get it high enough, a strong enough charge, if you don't understand these graphs are on the right, don't worry about it. It's just showing that we're getting a strong enough charge, enough positive sodium ions here at the axon hillock. If enough come and make their way to the axon hillock, we can get what's called an action potential. This axon hillock is a decision point. Are we getting enough charge here? Yes or no? It's a binary choice. We either do get enough signal and then we start something called an action potential, or we don't get enough signal and we don't do anything. Let me go back to that previous example of the strong person competition. If you can hit that target as many times as you want. If you don't hit it strong enough, you won't hit the bell and win your prize. You can hit it a million times and not get anywhere close near that bell. You're getting stimulated. Your neuron could be stimulated a million times, but it needs to be stimulated strong enough to reach that threshold. If we reach that threshold, our decision point, our integration center, the axon hillock, if we hit that strong enough, then we can reach what we call an action potential. So we go from a graded potential to an action potential. An action potential, this is all or none. It either happens or it doesn't. You can get graded potentials of all different kinds. Some sodium coming in, a lot of sodium coming in, but only once you meet a certain threshold, once you meet that limit, then you can get the action potential. That's what I mean by all or none. You either do get a signal or you don't. That action potential starts at the axon hillock, goes down the axon, and ends at the axon terminal. It's another flow of electricity. potential is referring to electricity. Action because it's a big action. It's very quick. So these graded potentials they lead to action potentials. They cause the action potentials if they're strong enough. At the axon hillock, we need a strong enough charge. The reason why we need a strong enough charge at this particular place is because we have special channels. Channels that are sensitive to charge. And if these channels are activated with enough charge, enough sodium ion coming in that area, then they open up and let even more sodium in. If you let even more sodium in, that lets even more charge come in, which means there are more channels over here that get activated because of all that sodium. So they let more sodium in, and it goes to the next spot. And there's more sodium channels that let more sodium in, and it goes to the next spot. We get this domino effect. Sodium channels open up, which open up more sodium channels, which open up more sodium channels, which open up more, on and on and on until you get to the axon terminal. That's why it's called an action potential. It's a lot of action going on, and it's all or none because once you set off one, the rest go off as well. Once you push over one domino, the rest go off. The rest fall over. So this is reliant, once again, at what's going on here at the axon hillock. If you can activate those voltage sensitive channels, then you've set off the whole thing. Electrical flow, those positively charged ions, they flow from the axon hillock to the axon terminal. Once we get to the axon terminal, we have vesicles full of neurotransmitter. We have vesicles full of neurotransmitter waiting at the axon terminal. They're waiting to be released. The electricity coming to the axon terminal is the signal for exocytosis. They're released by exocytosis. So we've gone from a graded potential. Let me redo this. We've gone from a graded potential in the dendritic cell body area, transitioning to an action potential from the axon hillock to the axon terminal. And that's all electricity. And now we're getting neurotransmitter release. And that's chemical. We're relying on physical molecules. Oh, I can't spell chemical. Chemical, there we go. So graded potential and action potential, this is electrical signal, very, very fast, while the release of neurotransmitter, relatively slower. Nice thing is it doesn't have to go very far. You're going over that really tiny synaptic cleft, traveling from the axon terminal of this neuron to the dendrite or whatever it is we're controlling on the other end. Travel a short distance in that interstitial space. Now we've mentioned myelin sheaths. What role do they play? What do you first need to understand about axons? Axons are leaky. You ever see those leaky hoses? The point of a hose generally is to have water go from point A to point B. You don't generally want to lose water, but let's say you have one of those leaky hoses, you're losing a little bit of water every step of the way. So the amount of water that you started with, it's not going to be the same as what comes out on the other end because some has leaked out along the way. All our axons made of plasma membrane, all axons are leaky. If you're curious why there are leak channels, that's something you're going to focus on in physiology. But you're losing positive charge. You're gradually losing charge. What myelin sheaths do, they cover up those leak channels. So if you have leak channels normally here, you're covering them up. So now nothing's leaking out. You're preventing anything from leaking. They act as insulators to charge. Just like blankets act as insulators for heat, they trap the heat that we have in ourselves. These act as insulators for charge. They're preventing any ions from leaking out into the interstitial space. So that's the benefit of myelin sheaths. But why don't we just cover the whole axon with myelin? If we did cover the whole axon with myelin, then let's say there's a sodium ion trying to make it down. Now we'd be relying on the physical ion to travel from one distance to another, and that can take some time. What's faster is if you do it like a relay race. You could run 400 meters by yourself, but it's probably going to be faster if 4 different people each run 100 meters, and you just pass off the baton. That's what's happening at these nodes of Ranvier. Let me clear this. At these nodes of Ranvier, these gaps where there's no myelin, you have a high concentration of voltage sensitive channels. So when the sodium ion reaches this area, we get a strong influx of sodium. Then the sodium only has to travel a short distance where it's insulated, and then more sodium comes in, and then it travels a short distance where more sodium comes in. We get a very jumpy type of conduction. Just like in a relay race, you run really fast, pass it off, run really fast, pass it off. That's what's going on here with the sodium ions. These sodium ions travel a short distance, activate channels, travel a short distance, activate channels. The term for this type of conduction, because we're getting this skipping, this jumping, is saltatory conduction. Saltar in Spanish means to jump. We're jumping, not literally, but it's jumping from node to node. So the nodes of Ranvier work together with the myelin sheaths to create this saltatory conduction. Not every axon has myelin sheaths and nodes of Ranvier. Some don't. If they don't have axons, if they don't have nodes of Ranvier or myelin sheaths, it's a generally slower conduction. If you've ever been hurt in such a way, like physically hurt in such a way that you get this slow pain, this slow, dull pain, those are because of unmyelinated axons. So there are some axons that are just normally not myelinated, or if the axon's really short, we have axons in our eye for vision that are really short, they don't need to be myelinated. The neurons that control our skeletal muscles, these have to travel a really long distance, and we want to control them very quickly. Those axons should be myelinated. There's a condition, though, there are conditions where we lose myelin, where we get demyelination. So normally there should be myelin, but then the myelin gets degraded for some reason, whether it's an immune system attack or some genetic problem or a virus. We start to lose our myelin. If you lose myelin, electricity leaks out. Those ions, that's what electricity is in this case, the ions leak out. The positive charge leaks out. And so you won't activate the next channels in line. And so you don't get any signal. Even though you're causing the action potential over here at the axon hillock, you're not actually sending a signal to the very end of your neuron. So I could be thinking, I want to control my hand, I want to control it, but if I have demyelination, I won't be able to control it. You're literally paralyzed. Conditions like multiple sclerosis involve a lack of control, and these can get especially serious when, if you remember, the muscles for breathing, those are skeletal muscles. If you can't control your diaphragm, you will suffocate because you can't breathe. So to end on a little downer there, that's where we're going to pause here for this video. On the next video, we're going to look more at the different types of neurons and how, again, that plays into input, integration, and output. We'll go over sensory neurons and then some integration and motor neurons. All right, please let me know if you have any questions. Thank you for watching. I'll talk to you all later.